Hello, everybody. I'm Silas, and welcome to Psilocybology. Let's expand our mind. This is a series where we talk about the research on psilocybin, the psychedelic prodrug found in magic mushrooms. We'll be breaking down and discussing the scientific literature, one study at a time, from past to present. If you'd like to support the show, please make sure to rate, review, and subscribe to the podcast. Today's episode is on the article titled Simultaneous Studies of Blood Sugar, Behavioral Changes, and EEG on the Wake Rabbit after Administration of Psilocybin, published in 1963 by researchers from Hebrew University. This is such a tangent, I apologize, but the title reminded me that for some reason it's a pet peeve of mine when someone says they're on a sugar-free diet instead of an added sugar-free diet. I realize that practically speaking, a true sugar-free diet is just not even an option, but hearing about so-called sugar-free diets has definitely confused me in the past when it comes to understanding what does and does not contain sugar. I know I'm not the only one. In fact, I once knew a patient with type 2 diabetes who kept getting worse and worse and didn't know why. He made substantial changes to his diet, cut out candy and a lot of processed foods, but it wasn't helping. It turned out that he was eating somewhere around 10 to 20 servings of fruit a day because he didn't know that fruit had sugar in it. I know that might sound obvious to some people, but if you were never taught that, it's not as if a lot of fruit comes with nutritional labels if you're genuinely buying fresh produce. Fruit is obviously healthy, but it still impacts blood glucose. So for someone controlling their diabetes, fruit needs to be eaten in moderation. I know I can get a bit too fixated sometimes on in quotes, properly categorizing things. But I genuinely think that it would be helpful if people more accurately describe their diet as being free of added sugar, or alternatively just saying they're eating a low-sugar diet, and being mindful that that would then mean they should be using portion control for foods like fruits and some root vegetables like carrots. So I know that was very far off topic, but let me bring it back to the study at hand. One of the things that researchers in this study essentially wanted to test was if magic mushrooms, despite themselves being low in sugar, might still cause an increase in blood sugar due to a physiological reaction induced by psilocybin. As background, there are two key studies that relate to this outcome. In episode 16, although the study we discussed was primarily focused on understanding the impact of psychedelics on color perception, the researchers of that study did also measure blood sugar levels in human participants 2 hours after being administered one of 3 psychedelics. They found no increases in blood sugar in response to either mescaline, LSD, or psilocybin. They did, however, find increases in circulating free fatty acids, which we saw again in episode 17. Increased circulating free fatty acids, as we've mentioned once briefly on the podcast, can inhibit the uptake of glucose from the blood, thus resulting in higher blood sugar. One of the reasons the researchers from episode 16 found an increase in circulating free fatty acids, but not an increase in blood glucose, may have been due to the fact that the participants fasted for 8 to 12 hours prior to the experiment. And I don't believe they were allowed to eat until the experiment was completed. That's an important distinction to keep in mind when we discuss the results of this study. In this experiment, the researchers administered psilocybin to rabbits, which did not appear to have been restricted from eating prior to the experiment, and were actively allowed to eat during the experiment. Now, although blood sugar was one of the primary outcomes of the study, the researchers also aimed to assess behavioral changes induced by psilocybin. They also recorded electrical activity in 3 brain regions to see if any changes in electrical activity corresponded to changes in blood sugar and behavior. In total, 15 rabbits were used. 10 were injected with 3.5 milligrams of psilocybin per kilogram of body weight, and 5 were injected with a saline solution as a control group. For assessing blood sugar, blood was drawn every hour, starting from the time of injection to 6 hours post-administration. For assessing behavior change, the researchers examined and coded for 7 specific behaviors. These were motor activity, food intake, water intake, response to whistling, response to flashes of light, response to aggressive hand movements, which were made in a purposeful attempt to scare the rabbit, And finally, whether rabbits expressed any behaviors that are apparently indicative of tension, which included moving their whiskers, chewing, licking, or crying. For assessing electrical brain activity, they implanted electrodes specifically in the frontal lobe, the parietal lobe, and the occipital lobe. Getting straight into the results, the researchers found that in the experimental condition, blood sugar rose and peaked within 2 hours after administration, and to return to normal between the 5th and 6th hour after administration. Overall, the rise was roughly about an 80% increase from baseline levels. No change was seen in the control condition, as expected. As we noted earlier, this is not the same response that we saw from episode 16, which found no increases in blood glucose. My hypothesis would be that the difference here can be explained by the fact that in this experiment, the rabbits may not have fasted before the experiment began and were allowed to eat food during the study. Although on that note, they found no differences in food intake between the control and experimental groups. Still, since we have seen that psilocybin appears to increase circulating free fatty acids, if the rabbits hadn't been fasting prior to the experiment, then any food that was being metabolized would produce a rise in blood sugar, since the free fatty acids would inhibit the uptake of glucose from the blood. So moving on to the behavioral measures, the only change in behavior the researchers didn't see in the experimental rabbits was, again, that there was no difference in food intake between the control group and the experimental group. For every other outcome, they saw some change in the experimental rabbits that started around 1 hour after being administered psilocybin and lasted about 4 to 5 hours post-administration. Specifically, the researchers noticed that the rabbits given psilocybin exhibited less spontaneous movement, a greater amount of water consumption, faster ear and head movements in response to both whistling and a flashing light, faster leg movements in response to aggressive hand-waving, and more crying, licking, chewing, and overall behaviors that indicated restlessness, and what the researchers termed emotional tension. Finally, the researchers observed marked differences in the electrocorticographic readings taken from the rabbits given psilocybin. Specifically, around 45 minutes after administration, they began to see what was so-called desynchronization and arousal. I want to note that the term desynchronization can sound a bit misleading. I read an interesting paper recently published in 2013 titled, quote, "Finding Synchrony in the Desynchronized EEG." And it made the case that what has historically been referred to as desynchronized EEG, marked by high-frequency electrical activity, should really be termed an activated EEG pattern. The reason early researchers characterized this pattern of high-frequency activity as, quote, desynchronized, is because they noticed that high-amplitude oscillations disappeared during states of arousal, and they associated those oscillations as a sign of synchronization. The problem with this characterization is that the absence of oscillations does not prove that higher frequency and lower amplitude activity is not also synchronized in ways that may just not have been as easily noticeable or detectable given technological limits of the time. So really, a better way to characterize the activity found in this study is that psilocybin induced an activated EEG pattern, because what they observed were increases in high frequency and low amplitude brain waves, which is the same pattern we've seen on previous episodes of the podcast. Now overall, when you look at all three of these outcomes at once, you essentially see that they all correspond with one another. The authors did a fantastic job illustrating this in what's probably the best figure I have seen so far out of all the studies we've discussed on the podcast. I'll include it on the episode transcript and upload it to the Instagram for anyone who wants to check it out. In essence, it just shows how the effects across the three outcomes really start to emerge 1 hour after administration, remain fairly sustained up to about 4 hours post-administration, and diminish by about 5 to 6 hours post-administration. Just as with the Siamese fighting fish that we discussed last week, I'm continuously impressed with how similar the time course is across different animal species. Anyway, the authors end the paper by stating that, given the pattern of effects seen, psilocybin administration appears to produce very similar biological responses engendered by adrenaline administration. When I read that section of the conclusion, it reminded me of two key pieces of information we've already discussed on the podcast. First, remember earlier how we mentioned that in the studies from episodes 16 and 17, the researchers found increased circulating free fatty acids? Well, as discussed in more detail on episode 17 in particular, circulating free fatty acids can be used as a proxy measure to assess changes in epinephrine, also known as adrenaline, because increases in adrenaline cause acute increases in circulating free fatty acids. The second thing that clicked for me goes all the way back to episode 13. During that episode, we discussed a study that tracked how psilocin, the psychoactive metabolite of psilocybin, was distributed throughout the various organs of rats. What they found was that one of the regions with the highest levels of psilocin were the adrenal glands, which produces adrenaline and is part of our endocrine system. Given these two pieces of information, it seems like this early research from the 1960s has already begun to elucidate the impact psilocybin has on our endocrine system, which can then have a wide-ranging impact on our metabolism, sensory perception, stress, mood, and other important physiological processes. At this point, however, the research we've discussed hasn't been able to determine conclusively whether or not this is a key mechanism of action that mediates or contributes to some of the principal phenomena induced by psilocybin, or if it's a less important side effect of psilocybin. I want to end the episode just by saying that on the bottom of the first page of this paper, the authors noted that this study was aided by a generous donation from Miss May Stein in memory of her late husband, Saul Stein. I have no idea who they are and couldn't track down any records to tell you more about them, since the only other note the authors mentioned is that May lived in Canada. But I wanted to acknowledge her contribution. I'd like to believe her and her husband would both be encouraged by the more recent revival of this line of research. I can only imagine what their lives may have been like. Well, that's it for today's episode. As always, I really hope you enjoyed the show. If you have any questions, feedback, or ideas for the podcast, please let me know. You can find out all the ways to reach me on the website, psilocybology.org, where you can also find the full transcript for each episode if interested. Thank you all for listening, and I'll talk to you next time.